Buenos días, ¿cómo están todos? ¿A quién tenemos de anfitrión hoy? ¿Hernán o Julio?
Buenos días, doctorado. Hoy estoy yo y está William de confitrión.
Perfecto.
Hernán nos va a estar asistiendo en la cuenta de YouTube, leyendo las preguntas del chat si hubieran.
Ok, perfecto.
Ujú.
¿Y quién fue hoy a
¿Me
los primeros?
escuchan?
Sí, hola
Escucha
William,
bien,
¿cómo estás?
Hola, hola, ¿todo bien? ¿Y tú?
Bienvenido a Confitrión GES.
Y yo estoy aquí chequeándolo por si acaso, ¿oyeron?
Bienvenido también, pero no se
Bien, ya.
preocupe que ese carro rueda solo.
Sí, yo sé que sí.
El
Eso
de
es lo
piloto
bueno.
automático.
Estos muchachos están encendidos.
Sí.
Están. Me
Lo
gusta
primero
eso.
que se activó fue el piloto automático, entonces ya después no hay problema con eso. El
Qué
vuelo no
bueno.
para,
Un abrazo a todos, que hace mucho que no lo veo a ninguno.
por
Abrazo,
suerte.
doctor. Un abrazo, doctor.
Malvado.
Doctora, hoy
Sí.
es Alex. Alex fue al GES el
Alex,
día de hoy.
perfecto.
Sí.
¿Ha habido alguna novedad, algún inconveniente, alguna notificación?
Nada que le hayan informado a Hernán, yo creo. ¿Sí? ¿Te informado algo,
Ok,
Alex, de algo que
perfecto.
haya pasado? Todo bien, dicen. Que ya habló con él.
Perfecto. De acuerdo.
Listo, doctora.
Buenos días, doctor Valle.
Hola, ¿qué tal? Hola, Hernán. Bien.
Buenos días, doctor. Le habla Julio. Aquí estamos de anfitriones hoy con William.
A ti te oigo lejos, Julio.
¿Me escucháis mejor?
Ahí mucho mejor.
Ah, perfecto. Tengo que acercar el micrófono mejor entonces.
Este webinar tiene mucha demanda, mucha popularidad.
Sí, lo tenemos transmitido vía YouTube, así que el que quiera verlo por YouTube, ahí está el canal compartido en las publicaciones que hemos hecho. Ya está la transmisión en vivo desde YouTube. Ya el que entre a YouTube en este momento puede verlo directamente en la página de HES Academy en YouTube.
Hola, Friné.
Hola, doctor, ¿cómo está?
Contento y bien.
Me alegra, así lo veo.
Ajá.
Qué bueno.
Chief, ¿usted oyó un audio que le mandé ayer?
Déjame ver si lo tengo aquí. Yo me voy a enfermar de tantas cosas que yo... ¿Y era personal o era un grupo?
Filtre, doctor.
Fue personal, doctor. Yo pensé que usted me tenía cariño todavía.
Ah, imagínate tú. Lo del cambio de horario.
Hola, Chief, ¿cómo está?
Sí, mañana, el viernes, lo de Elena Barraquer.
Ah, el de Elena Barraquer.
Sí, es el viernes, doctor.
Sí, yo
Una de
le
la
mandé,
tarde.
va a ser a la 1 de la tarde. Yo le mandé un audio, doctor, para usted.
Y eso lo tengo yo con altísima prioridad.
Pero oiga el audio que le mandé, no se ponga así, porque usted se pone así conmigo. Después usted lo oye ahorita.
Ok. Activo, como dicen los Boy Scouts. Activo. Mande usted. Mira, por
Julio,
cierto, que a Mena y a Maldonado y a mi grupo mexicano, me metí en el espectáculo este de Escaré del domingo. Eso fue algo sensacional, pero lindísimo. Lo vi enterito. No sé si hay algún mexicano ahí que tenga micrófono, pero eso fue...
Sí, aquí estoy yo, doctor. Sí, no. Es un espectáculo, increíble eso.
Sí, yo lo vi
Porque
también, fue espectacular.
hasta la conquista y luego van yendo así como a diferentes sitios del país y pues ya demostrando sus culturas, sus bailes y los cantos. La verdad es que sí, es un show que está muy, muy bien hecho y muy bien planeado.
Qué bueno. Bueno, yo veo que ya también llegó al aula virtual el doctor Ike Ahmed. William va a estar como coanfitrión. No creo que ahora tenemos, todavía faltan 5 minutos, o sea que creo que ya podemos hacer el cambio a inglés, que es el idioma que vamos a manejar en el chat, ¿verdad?
Hola, William. A todos, ¿cómo tú estás?
Todo muy bien, doctor. ¿Y
A
usted
ver,
cómo se
¿quién es que
siente?
le va a dar la bienvenida? Muy bien, ¿quién le va a dar la bienvenida al doctor Ahmed?
Julio va a hacer una introducción y luego yo paso. tengo una pequeña presentación y luego ya él comienza.
Okay.
Esperamos a las 11.
William, dame un favor, dale la vuelta como estás sentado para que la luz te dé en la cara. La luz está dándote desde atrás, entonces te empaña el lente.
Perfecto.
Ah, claro, mucho mejor. Sí, claro, y con arte y de todo. No, no se ve.
Muy buen fondo. Bueno, voy a poner mi micrófono en mute para reducir el ruido en el ambiente.
Vamos a poner todos los micrófonos de las...
Entonces, el que molesta. Bien. Parece que hay un webinar, William, también de Juan Carlos Izquierdo, de Perú. Y él me estaba pidiendo que participara. Y hay una serie de cosas. A ver si nos combinamos tú y yo. Porque es de glaucoma, ¿no? Ya.
Hola, hola, everybody.
How are you?
I'm good. How are you doing?
Terrific. So good to have you.
Good
Morning,
to see
Ike.
everybody. Juan, looks like you're always working.
Well, I got my Paco Caribe cap on.
Boy, that was my last meeting before everything got shut down.
It was amazing. And so happy to know that Lucho that came out of that infection without a problem.
Wow, that was very scary. I mean, you know, we know the stats on that, so it was great to see him back, and he's right back in there working.
Hi, Ike.
Hello, hello.
So we
Carlos Gomez.
we have an 11
Carlos.
at 11. We're having our faculty and residents.
I think you're muted, Juan.
Yeah, I think he
and fellow take over from now on.
Hola Juan, hola Carlos. Hi Ike, how are you?
Hello, hello.
How do you do, Carlino? How are you?
Hola, hola.
How's it going out there?
Very nice. Waiting for you.
Terrific. We're having good weather. And we've had 286 people die, so it's been a reasonable result. We're not
Okay.
over the hump yet, but it looks like it's turning now.
Yeah. I think it's likewise here in Canada.
Mhm.
We have a couple. You know, our big cities tend to have more clusters,
yeah.
but our curve is definitely.
We still have towns in
Sorry.
the virus that they can start from zero again. So we're reopening.
Sorry, Juan, I couldn't hear it. I couldn't hear you completely. I don't know if the connection. Can you guys hear me? Okay.
Yeah, I can hear you. Okay.
Yeah, I can hear you.
Okay. Yeah, I got a little bit. I just missed a bit of that transmission. But yeah, no, it's good. It's, lots of webinars.
I think he has a glitch there.
No, you have to be selective almost not to.
It's very true. It's like getting invited to too many meetings. You have to kind of decide which one not to go to.
Yeah.
So what's the first
right
today?
when you go to one of those Asterisk meetings or the Academy, and there are like 30 lectures all at the same time, and you have to decide which one to go. And then if you go to one, you miss 10 others, and you say, you feel bad about it, but you can't.
I know. It's very true. Very true. It's very true. It's a different world. Hey, just for the program today, I have a lot of slides, of course, and I'll go through that. But did you want to be able to take questions and things during the presentation?
Yeah.
The format has been that the host, and in this case, your host is Dr. McCollum. He'll be going on with things, and he'll be fielding the questions from the chat room. And obviously, if something is not clear, he'll have to chip in and say, you know, we missed that or we lost connection, help you out. But we should. The group is pretty good about using Zoom, and we're very interactive through the chat room. But that way you don't get interrupted. So you can
No
just
problem.
walk.
I take a peek. I've learned to take a good peek at things when I need
Okay.
to. So
Yes.
so no problem.
Perfect. So, Julio, if you want to start.
Yes. I just want to say good morning to everyone. Good morning, Ike. This is Julio Navarrete. I'm a third-year resident. On behalf of the residents of our hospital, we just want to welcome you and thank you for being here, taking the time to
Keep the microphone close to your mouth, Julio.
Yeah, thanks, Dr. Valle. Yeah, we just want to thank you for being here with us, speaking about innovation and research. You're one of the most renowned ophthalmologists in the area, so we're very pleased. The way that the class is gonna go is we're gonna let you talk, go through the slides, and at the end we're gonna read the questions on the chat, and on the chat on YouTube. Hernan Bejar, our third-year resident chief resident, gonna help us with those questions. So I'm just gonna go ahead and turn off my video and my microphone. I'm gonna let William take over from now.
Great.
Okay,
Thank you for the invitation, guys. Good to be here.
Perfect. Let me show my presentation. Okay. Perfect.
And
can you see my?
You got it.
Can you? Yep. Perfect. So perfect. So good morning to everyone. Hi, Dr. Ahmed. Good. Thanks for your time, for being with us today. Today we have with us Dr. Valle, who is Dr. Juan Valle, who is our chairman, Dr. Frines Salazar, that is the academic coordinator, and the residents and attendants of Hospital Dr. Elias Santana in Santo Domingo, Dominican Republic. We are also live on YouTube for everyone that wants to join this meeting. So today our lecture is Innovation and Research in the Ophthalmology Practice. Our speaker is the incredible Dr. Ike Ahmed. Let me pass the presentation. Okay. Dr. Ahmed is someone that really doesn't need an introduction. As we all know, Dr. Ahmed is part of the very prestigious list published by the ophthalmologists called the Power List. Is the division head of ophthalmology at Trillium Health Partners, Mississauga, medical director of Prism Eye Institute, director of the GAS Fellowship, assistant professor of the University of Toronto. Also, I want to mention that I had the opportunity to see by myself what is behind all that success of Dr. Ahmed, and that is really hard work, dedication, and the most important thing is admiration and respect to give his patients the best care. Great, Dr. Ahmed, you can share your screen.
Thank you, William. Well, I just want to say that it's a pleasure to be here. You know, I was in Dominican a couple of years ago, Dominican Republic, and it was a wonderful time. And you have just great folks here. Of course, Juan Valle is world renowned for his work and his knowledge and his sharing. And I realized that actually he is well known for his dancing skills. And I witnessed it numerous times, especially when he's in the middle of a pool with all the water going around. I noticed that he is, you know, very good with the moves. So he's not only good in the OR, he's actually really good in the water as well, dancing. So it's always something to watch and enjoy. So I'm going to just share my screen here, and William. Oh, I guess I need to. I guess you need to maybe stop sharing here because I can't share my screen. It says,
Yep.
There we go. Okay, perfect. All right, so I'm going to share again here. Thanks again for being here. I'm going to hopefully speak to you and have some nice interaction with regards to doing research and innovation in practice. I hope you can see my slide there.
Yes.
And so let me just quickly do that. Okay, good. All right. Just want to share a couple of things on social media that we're starting here so people know. Yep. So this is something that a topic that is really, really dear to me. When I started practice, I actually went into private practice and I wasn't in an academic environment right away. And so I missed that aspect of practice. And so really, really starting with nothing, you know, built into an organization that I think we're doing some hopefully good work. And I'm going to speak to that a little bit. I know William had asked me to just say a few words about how things are going here in Toronto and Canada. This is our city. William lived just nearby the CN Tower. He was our downtown guy, very personable, as you all know, very affable. And William was quite integrated with our research team and was very productive and great to work with. So in Canada, we've been on lockdown for the last 6 weeks. Elective surgery has been stopped. I have gone in to do urgent glaucoma surgery a few times throughout the course of the last few weeks. And our clinics are basically shut down. So it's really a dramatic change in of course, how we're used to doing things. I'm sure like it is in Dominican as well. This has given us time to pause and reflect during this challenging time. And it's a time that I think all of us are just figuring which way to make the best of it. It's really brought us all together. I think, I can't think of any other time we've been connected with the world, although it's not in physical, it's certainly, in virtual fashion. And that's says a lot as far as the ability for humans to persevere. Prism Institute, which is where our clinic, as William knows, it's a brand new clinic we built. And it's a big facility. We have a big research department, and now we only use maybe 5% of the clinic because we shut down, as you see, and we just have a small area that we're working, which is just only where we see urgent patients. And we've moved to virtual care, which has been quite an interesting journey for me, because I'm predominantly a surgeon, to be honest with you, and I love the OR, and that's my area. But now doing virtual work and setting this up has been a whole new opportunity for me, and I've actually looked forward to it, and I see brighter days coming up in our ability to move on to this as well. And I think the ability to do what we're doing now, which is to do learning and share. What an incredible opportunity, and I hope everyone is using this and leveraging it as HES and the folks here on the call, being able to kind of work together, share together, enjoy together. Social media is a buzz, and it's really it's an incredible link that we've seen here as well. And as we talk about this topic, of course, I apply the knowledge that I've made over the years in reading and learning from others, being in the OR and following folks. And I think all of us do this as well. And I think one thing that I just want to start off saying, and I spoke to my Brazilian colleagues a couple of days ago, and I just said a couple of things about the fact that don't underestimate what you bring to the table. We're going to talk about science and evidence and this and that and follow the protocol. But the best, part and ingredient of what you're going to do in terms of research and innovation is really what you bring yourself as far as the human factor, what you bring to the table you can add to this as well. And just make sure we, of course, check our ego at the table, because ego gets in the way of science and it gets in the way of progress. And it's fair to say there's probably a lot of ego around when it comes to research. Everyone has their own ideas they want to push. And we can often get on the wrong path if we let our ego take over, which is a natural thing for us to do. So try to be careful and mindful of that and bring the spirit of what humanity is about. And just remember that, we're all going on this journey. And this is, for me, a journey of exploration with research, a journey of thinking differently with innovation. We want to make this journey count. And that means being an observer, taking in everything, trying to understand the chaos in the world and trying to organize it and being receptive to everyone's ideas. And I think the team that we have with us, with our trainees, our research and fellows, our research staff, our clinical fellows and residents on all do a lot for this. So let's just talk about innovation and research. I think a lot of people confuse the two and they think it's the same. We think it's the same thing, but really research and innovation is different. Research, I would say, is a specific question we're trying to answer to gain knowledge about a particular subject, particular topic. When we do an experiment to get that question or to answer that question. Very much important to be able to ask the right question when we talk about research. And we have a certain framework we work within. You have to be, of course, somebody who is good at asking questions and trying to look for what the problems are. On the other hand, innovation is a little bit different. Innovation isn't necessarily doing a specific experiment for research. It's about looking at a system or looking at something and finding a better way to do something, better way to do it, a better way to do surgery, for example, or a better way to have the flow of patients, how we manage patients during COVID. And for this isn't necessarily a science experiment. This is not necessarily asking the right question even. It's thinking differently. It's thinking differently. It's thinking, looking at things and problems in a different way. And of course, there are some areas that cross over, but it's important to differentiate it. And some of us are really good at research. We can sit down in the lab or sit down in a study and just, you know, have the paperwork, have everything organized and design the trial with the right questions to ask and know the right things to measure. Well, some of us are not necessarily that into that as much, but more into the, you know, hey, listen, how can I change the way I do something? How can we do it better? How can we make the experience better? Whatever it may be. That's the applied way of how we can take research to perhaps a more systematic or commercial way. And as far as in the commercial world, this is kind of how they look at it. You know, you need to basically gain knowledge by doing research. You need to fund research with money. And you need to basically then answer the questions. Once you have some questions answered, you can then innovate and implement it into a system or commercialize a product. And that's where, and then you make money. I say you in terms of, let's say, a company developing something. And so that's an important way to distinguish the two. I don't know who's an innovator, who isn't. I mean, we often talk about people who are innovators. I don't particularly think of myself as an innovator, but I do think of myself as somebody who stays close to the ground. And that's why I like to kind of, when I talk about innovation, I like to talk about staying close to the ground and being really close to where the action is, to have, you know, an ability to really just look deeper into things, but not from a very high level. I mean, yes, we need to look at it from a top-down way, and then you need to do that to zoom out and zoom in. But I'm not a very sophisticated person, as William will tell you. I'm a pretty simple guy, and for me, it's straight up raw, you know, skills. You know, anything we do in this space really starts with ideas, and it means we have to be able to dream, we have to be able to imagine, we have to be able to visualize, and the ideas are really the basis of creation, of life, of creation, of new ideas. And so, really, we have to start with this thinking here. No idea is dumb, of course, but there are a lot of dumb ideas out there, right? So we have to start with an idea. We have to know, of course, which one develops or not. Now, there's a certain attitude, I would say, that I think innovators have. And, you know, we often talk about innovators having great ideas or they're skilled or anything along those lines. But I would say that, you know, great innovators are not necessarily, you know, skilled or necessarily intelligent, but what they are, they're resilient. Like, they're tenacious, they're flexible. You know, they can adapt. And that's the test of an innovator, is to basically work against the indifference that you see out there, right? You know, hey, listen, I think we should do glaucoma better. Well, you know what? The community doesn't really think so. There's a lot of indifference, apathy. You know, do we need to do that? What's the point? And I think that's the challenge, the biggest challenge of innovation in an innovator, is dealing with that type of being shunned and being ignored in the indifference. And being ignored is difficult because you need people to kind of respond to what you want to do, or the society or the people you look up to. And so that's the resilience parts, which I think we have to really build into our quality and our character. You know, innovators don't mind failure. They don't care what people think, but they're willing to work damn hard to get it done. And I think that's the attitude that an innovator, I think, needs to bring to the table. And I love Alan Kay, you know, a very prominent computer programmer, who's quoted as saying, you know, the best way to predict the future is to invent it. You know, that is obviously the best way to think about what's going to happen in the future, is to actually invent it. So that's what you want to do, and be the first as well. And I think one thing that innovators have, which is, I think, a good quality, sometimes a bad quality, is that they are a little different. They're a little different. They may be quirky. They may be a bit of an outsider. They may be shunned, like I said. They may be called crazy. That's probably not a bad thing if you're an innovator. And so it's okay to be in that way. And for some of us, you know, we always feel that way no matter what. But I think that skill can help, like I said, to think differently. And, you know, I think that we all want to follow our idols and our mentors. We want to follow our leaders. We want to follow our preceptors. And I think that's the way to learn. But we have to know that we don't just want to be a follower. We want to basically find our own path and not be afraid to go there, but build the knowledge and the basis and the foundation to do that. And not only that, but put yourself in the right place. Work with the right people. find the right place to do this and find the place to be there as far as the right time. I mean, that really puts you into the best position that you can be. You know, I often say that, you know, people walk into a room or look at an idea, and some people are able to grasp it, others don't. And I think be open and receptive. You know, be willing to think about other ways to do things, and open your senses, your hearing senses, your visual senses, your perceptions. You know, I think that one of the biggest challenges is not being able to take in something when it might be right in front of you because you're not thinking about it, you're not looking for it, and maybe your mind is a bit closed. I think some of us are annoying because we question everything. And it's annoying. I'm sure it is, but that's how we make progress, because we then realize that maybe everything isn't true. And I'll speak a bit about that as well when it comes to research as well. And there's so many things out there that we think is dogma, but it isn't. And I think innovation involves the ability to question and not assume things at all. And I think one of the things that I think is a good quality to have is the ability to say, I want to be it first. That's a driver. We have to be motivated to be innovative. And I think one of the things that motivate some of us is we want to be the first person. And if we're not the first person, it's kind of a bummer, you know. And that's a driver to get things done. And it may sound kind of funny. I don't mean to be egotistical. Some of that, I guess, is helpful in that way, but it's certainly a driver. And I think that the definition of failure is not actually losing. It's basically refusing to get back up again when you fail. Because we all fail. We all fail a lot. We fail more than we succeed often. But I think that's, again, a quality that I think a lot of people who've been able to be successful have been able to do this in a way that is, be able to get up and go. And I think we also have to know and be aware and be willing to, not only do that, but understand and be willing to break the rules. Now, of course, you know, rules are there for a reason. So I'm not saying to go and socialize right now and go and hug everybody on your street and go outside. But I think what I mean by that, of course, is that we should challenge rules and understand why they're there and not make an assumption. And I think that rule breakers often are able to be successful because, you know, they do it in a thoughtful manner and a proper scientific based manner. The collaboration is really important. I mean, that goes without saying. If we can't collaborate and have partnerships, it's very difficult to do innovation and research in isolation. And so find the right people to work with and use your network and build that partnership, which is really important. You know, studies like these where we basically, you know, did major publications in clinical trials only happened because of collaboration and knowing our network. And that, I think, allowed us to build and to hopefully produce high quality work. So use that. It's a global village. And now's a great time during the quarantine to be able to connect. It's also important to know the market out there. And I'm not talking about the stock market. I'm talking about, you know, the medical clinical outlook on things. What's going on? Who's doing what? What do people want? What are the needs? What are the interests? What's the direction going in? And no matter what, we know we have to work with industry because they're a partner here. It can also be a challenge and a conflict as well. And so it's important to ensure that we differentiate what our goals are and we come together on the goals that are common. But without working with industry, it's very difficult to be able to move forward beyond simply just an idea often. And I do consult with many different partners, and I put my disclosures up, you know, all the time, every time I speak, because we know that, of course, this can be a conflict, and we need to challenge those speakers that are conflicted, whether it's financial or otherwise, because that can certainly be an unintentional bias. It's very, very important. But I do want to say that partnership has been of value to me. We got to keep on moving. We got to keep to keep on running and not stay put because you get stagnant. And status quo means you're falling behind. So innovation, again, is about not staying put. Now, we don't want to innovate for the sake of innovating. And that's also a mistake sometimes people make as well. But, certainly staying put and saying this is a final solution here. There's always a better way to do something. And no matter what the talent is or what you think your talent is, I can tell you from my experience working with so many people and my own personal experience is that hard work and the attitude trumps talent, by miles. And so don't feel you have to have a particular special talent, but if you can work hard, man, you can do a lot of great things and be an innovator. I love Muhammad Ali's quotes, and I think I have a few quotes on my wall, but I often like this quote about impossible is nothing. And if you just read the quote on the bottom of the screen there, it's a word that people often use, people who are insecure, people who are in positions of power, and people who want to keep in that position. It's a word that's thrown around a bit, but if you want to change the world and you want to be able to make a difference, then we need to challenge this. And impossibility is not a fact at all. It's an opinion, exactly as Muhammad Ali said. So think about that. Impossible, not a final thing. So that's a bit about research. I want to get a bit more into the granularity. Innovation. That's some ideas about innovation. I want to get a bit more granular on research and speak a bit to this aspect of it. And again, as I said earlier, like, what does research mean? It means different things to different people. Many people here, I'm sure, are clinicians. They're not necessarily, their career is not necessarily built on only doing research. Like myself, my career is clinician, and I guess a wannabe scientist. But I like what's listed here in one of the buildings in the Library of Congress, the Jefferson Building, where science is organized knowledge, right? Science is an ability to build our knowledge base, and give us answers to questions that are there, and I think that's the success of science. It's not knowing it. It's basically seeking that knowledge and seeking the ability to grow that data set, so to speak. That's what's important. Here's a quote: "Research is to see what everybody else has seen, and to think what nobody else has thought. And so that's the way to think about what research is about, what maybe other people really haven't thought, even though we've all seen everything here. And Johari's work window here, which I find always a cool thing to think about. And Donald Rumsfeld, Secretary of Defense back in the Bush years, popularized the phrase, the unknown unknowns. You don't know what you don't know. If you think about this pictorial here, and you think about, for example, the known knowns, these are things that we know, and we know they're true, and we know that is fact, and we've proven it. We also know that there are unknown knowns, and I'm going to give a quote from Mark Twain here, that it ain't what you don't know that gets you in trouble; it's what you know for sure that ain't so. What you think you know, but actually isn't true as an assumption. And that's the unknown known. So the unknown known is those things we think is true, but they aren't. That we think it's a fact and they aren't. That's dangerous, of course, because we can be very wrong and we think we're right. Known knowns are things that we believe are right and are correct. Known unknowns are things that we don't know the answer to, but we know we need to ask the question. We know we don't know this, for example. And then there's the unknowns, which are probably the most difficult thing to conceptualize, which is we don't know what we don't know, right? We don't know what that is. So, and that basically leaves us into this position here. So I love this. Again, I love this quote from Mark Twain, like I said before. And of course, in this day and age, more than ever, and certainly facilitated by the internet, we have the issue of fake news. And anybody can say fake news these days. And we know certain people use it more than others and on certain issues. And that's the challenge we all have here as well. So maybe we'll switch and talk about, why do we conduct research in clinical trials? Well, there's a lot of reasons to do this. Sorry, I didn't change that slide. My apologies. But there's a lot of reasons why we do it. Some people do it for the prestige and the credibility and the access to more, opportunities. Intellectually, it grows. It provides a leadership position. There's financial benefit potentially and new opportunities. I think we have all the reasons we do it, right? But I think maybe the most important reason we do it is because we want to answer a question. And a number of years ago, I remember in having a patient who brought this to me. This is basically a patient website where patients can talk about problems they've had and things. And this particular website, or this particular post, was about iridotomies, and it garnered a lot of posts, a lot of people commenting on it, patients, I should say. And what I'm basically writing here is that patient had iridotomy for angle closure or angle closure suspicion. I think we do less now based on, for example, the ZAP study. Nevertheless, we do them, and basically after the iridotomy, the patient had all kinds of problems. Some patients were so distressed that they were thinking about taking their life even. And I was shocked to hear that. If it's true. And again, it made you want to think, is there a better way to do something, and why is this happening? Well, going to the basic science of it and hearing a couple of folks that were, talking about why these phenomena occur. And the reason why people were upset was because of dysphotopsia, I should mention. And the proposed mechanism is basically was basically felt to be this base up prism that occurs when light strikes the base up prism that the tear film creates below the upper lid, and bends into the iridotomy site and strikes the peripheral retina. And that then potentially gives a patient bad glare and dysphotopsia. It's not common, of course, but it's enough of a problem for certain patients where, of course, it can be quite distressing. And so we did a trial. Very simple question. Can we put the PI somewhere else rather than superiorly? Because even if it's covered by the lid can be dynamic. And of course, by refraction of light, one can get light going through the iridotomy. And so, even though it's covered. And so we did this trial, temporal versus superior, intra-patient comparison, one eye superior, one eye temporal. Very simple study. It doesn't take a genius to design the study. We went through the proper consent process and everything else. And lo and behold, we found the results. And we found that we went temporal or nasal with the PI. We ended up seeing less dysphotopsia. And this was great. And we published it with my fellows, which is a great part of working together. And I think it changed a lot of people's minds about this. And it just, again, is an example of how we did research. So there's different ways to think about research, basic versus applied. And often we talk about basic science and clinical research, and that's a little bit different. Basic research is to basically understand how do things work, right? How does this thing actually function? It can be often at a laboratory level, and there's not necessarily an applied clinical level at that point. Well, applied research is now building on that basic science research and now saying, okay, in the clinic, in practice, these technologies or this particular work or this particular surgery, for example, results in a clinical result, and it solves a problem in the real world. And so, one can think about medical research in both of these ways, and we might all have different interests. Many of us who are clinicians are not necessarily in a position to do basic research work, although we do, but more on the clinical side. And there's different levels of research and different ways to do research. Registries are easy to do things. Major clinical trials, of course, are more work, and they can start from first in human work. And Juan has been instrumental in some of these early areas. But they can also be phase 4 studies after a device or a product, for example, has been approved, or a drug. We can do reviews. We can do meta-analysis. These are all ways to think about how research can be done. And when we talk about developing a study, we need to think about, is this a study we're going to do on our own site or multi-centers? It's obviously great to collaborate. We get more recruitment. We get bigger sample size, which is very important to think about doing a study. And then we need to think about, are we doing it ourselves? Is somebody sponsoring and coming to us? Is it a government nonprofit that's sponsoring it, or a hospital institution? These are all often where these studies develop and go forward. We hopefully know the levels of evidence in terms of what we're trying to do. It's not a problem to publish a case report. It's a great way to start, but the evidence, of course, is not strong, or the level of evidence isn't strong. On the other hand, of course, an RCT would be prospectively would be ideal, but it's difficult often to do. And putting together a case series or a case control or cohort would be good ways to get to that highest level, which is still valuable. There's nothing to be ashamed about by publishing retrospective work. I think as long as you account for subjects and you do it right, and you account for biases that can occur, it can be very valuable. So don't discount that. And I think there are some RCT snobs out there who only want to look at RCTs, and RCTs have their own problems, of course, as well, in terms of how they apply into the real world and the assumptions that they make. So every study has biases, and we know levels of evidence, of course, are important, but don't discount other kind of studies. All of us here, I think, have a different level of research experience, no doubt, and that's understandable. I think that those that are starting research, can do this on their own, self-initiated. Anybody can start research. Again, like I said before, being part of a larger trial is a great way to start. Someone's already done a lot of the work. They've asked the question. They've designed the protocol. They've looked at consent and forms and everything else, and you can join them as long as you have the ability to do it and recruit, and as long as you have research facilities to do that properly and people are trained. And of course, industry is a big sponsor of this these days. And again, it does create a conflict and a bias as well. So there's a lot we can think about. And I hope that the mindset is really what drives a lot of this, because I think you don't necessarily have a lot of research experience, but the mindset is what's important. There's a lot of reasons to fail, though. A lot of reasons to fail. Lack of ingenuity, lack of science, unorganized. You don't have the resources to do it. You can't execute it on a great idea, or you may lack the passion, amongst other issues. So identify these potential barriers and hurdles and deal with them. It's essential to have certain things, though, when we do research. And that's, of course, every research needs to have a protocol. We can't just go out and just say, I'm going to do something and not write it down and not organize it properly. We need to document research. And that means ensuring we have the right case report forms and design the right spreadsheets to collect data. We need to make sure we get proper consent with patients, and we have a proper consent that's written in the right language and approved to use. We, of course, have to have a budget that's built up for this, and ensure we account for the extra visits or the extra work that's done or patient reimbursement. RRB is essential for these things, and ethics approval is essential for any human subject study. It's important to get this, and if you're doing retrospective study, still apply for REB and maybe get a waiver or get approval. But do not publish or embark on a research study that involves human subjects unless it's a quality assurance project or a registry without getting proper ethics approval. And as an organization, it's very important that legally things can get upside down. And it's very important that we need to make sure we establish the right legal framework if we're working, for example, with sponsors, if we're working with industry or other universities, because this is where, even though things start great, like a marriage contract needs to be done, I think there are problems, then the parties involved can get quite upset with each other. Resources, of course, are always a problem. Research, unfortunately, involves resources and money, and that's often a barrier with this. And that's something that we have to always try to be creative with. And I realize the challenges with that. Research is not done in isolation. It's part of a team. William has been a great part of our team, and I know we'll continue to do that as he's back home in Dominican. It helps, and it depends, of course, on your size are. When I started doing research, I had nobody to work with, nobody. And we cross-trained people to help to do some work. And eventually we brought on people part-time as a research scientist, and then we brought a manager in, we built a team around it and everything else. So this is some of the people that are on our team: research managers, study coordinators, technicians that do some of the technical work, business manager or a business person to look at the business side of it, legal expertise and advice for legal contracts. We have trainees involved. I mean, research is a big part of our trainee work. And also we have to develop the right system in place to make sure, that, the system and the clinic and the institute is properly designed to do research and set up properly as well. Again, as I said before, one of the biggest challenges in setting up research is, especially if you're on your own, is the resources required. And of course, those of you that are in an academic environment have an advantage in some ways and a disadvantage. An advantage, of course, you have people already there. You have the facility, you have staff that can do this ideally and are trained to do this. The downside is that you perhaps it may be difficult to be mobile and agile, and the process may be slower, and there may be more hurdles to jump over. So in my setting here, I'm primarily, in a private clinic. We see public patients there. I also work in public hospitals for the most part, and also at the university. So I'm all over the place. But setting things up initially is a big challenge, because research is a part-time job for you, and it's like a startup. And so therefore, having staff, maybe an interested technician or administrative staff who's interested in maybe doing something more, can be helpful to be the contact person to do research or document things. And as you grow, that can grow. A technician is a great person to start with, because they know ophthalmology already. And if they can take an online research course, they can understand the basics of good practice. The mindset's important. Everyone in clinic needs to think about every subject as being a research. Every patient that walks into my clinic, I really tell the team that they potentially could be a research subject, with the permission, of course. And patients know now, coming into our practice, that potentially we may think about doing some research. And certainly patients understand that, and we need to explain to them the benefit of that, and also that they don't have to participate if they don't want to, of course. It's important to have space. I mean, in our old clinic, we had very little space to do research. Now we have a big space. And again, I talked about staffing and trainees and technology as well. There are certain things that we need to have, and there are quality standards, international quality standards like GCP or good clinical practice. You need to have a standard operating procedure depending on what research you do. Now, if you're just doing some case reports or some clinical or some case series, these things aren't necessarily as critical or not mandated. Although I do think they're good. But having actually like a protocol, being trained in the right international standards and GCP and SOP, and having SOP, I think is really important, particularly if we're doing major trials. I mean, it's actually mandatory if we're doing clinical trials. The funding part is always a challenge, and it's drying up in many parts of the world, and that's of course a big impediment. And the government's a source, societies are, foundation and donations from patients and others are helpful, but industry has been a big player in that now, of course, and they have helped to support industry directly and indirectly, and help research institutes and practices do that. And a lot of research now is being done outside of the typical academic corridor, and done in private practices. So we do see this kind of moving. Industry, of course, like I said, has a role for that reason. They often have common interest in the products that we work in, the patients we see. They often provide opportunities for us to apply for research. We call those investigator-initiated trials, or IIT. And they are involved in some of the big registration trials for drugs and devices. They also do marketing studies, what I call marketing studies, where basically it's a study, it's not a real study. It's just to get the product out there, and we'll collect some basic data. Those are not ideal or great, of course, but they may at least give you access to product. They may allow you to kind of get some early experience, which is fine, but we must call those out for what they are. They are not real studies. They're really marketing studies. And I think we should differentiate between evidence-based medicine and evidence-based marketing. And that's, again, a healthy skepticism for industry, which is always important as well. Again, when we get to the nitty-gritty of research, it always starts by asking the right question. And I see so many people make this mistake. I see it reviewing papers on editorial boards, or in seeing people at meetings and presenting. And I see people in our own clinic where there's an idea, but the right question is not asked. And that's always a challenge. And if you don't ask the right question, everything else becomes a problem. So ask a question, delineate it well, don't make it too big, narrow it as much as you can, and ensure that you're going to be able to answer it properly in the way your study is being designed. And as I said before, so as I said, define it, be specific, avoid generalized objectives. I want to see if this cures a disease. No, ask a specific question you want to know. Again, for the same reason, try to find one main objective, maybe two, and define them. Have one primary objective, main outcome measure. And again, publishing papers, we need to define this and define it clearly what it is, and build your sample size calculation on that. Any prospective trial comparative trial should run through a proper sample size calculation. Know how to do that, or have a stats expert to help you with that, because that is really important to ensure the study is powered right, meaning that there are enough subjects in the study to be able to reject, for example, a null hypothesis, or to see that there's a real difference statistically. So be sure to define things really well. We have to know the literature, know what's published, really do an in-depth review, and get your team to help you with this. Speak to people who've done research in your field for ideas. They may know the literature in areas that you may not know. And be a peer reviewer. Review papers, a great way to learn and know how papers are written. And I think one of the best places to know how to write a paper is to read papers. I mean, what a simple thing. Read a paper and see how they did their study, see how they wrote it. I mean, and there are a lot of copycats out there, but there's a reason that we can review and see how things are done. Chances are that it'll give you a good guide to see how it is. Now, don't follow everything, of course. Develop your own methodology, but it is something to think about. Being on the other side in terms of reviewing papers, reviewers are going to want to ask a few questions when you submit your paper for publication, which of course many of you want to do, and that's certainly an ultimate outcome for our work. Is this an important subject to study? Is it worthwhile studying it, or is it not really important and people aren't interested in it? Unfortunately, there are things that are important. Maybe people are not interested in it, and that's a sad reality. But that's something that reviewers think about. Is it valid? Is the study valid? Is the validity appropriate? Did the results support the conclusions? Did the methods properly produce the results that can be valid? Very important that we look at those things. And then again, can this study be generalizable? It is something that we can see that can go and be applied in the, to all patients or to all practices. Important things that we think about here. There are many errors we see in publishing. This reminds me of my computer when I drop something on the outside screen, and it's really feel handicapped not having a laptop. And now I have a backup, which is my old laptop. But there are a lot of errors that happen with publishing, and again, I mentioned some of them already, right? But statistical problems, of course, exist. A lot of biases can exist, and the way it's written and making grandiose conclusions can all be problems, and can really hamper the ability for a project to be published. When it comes to recruiting, I should mention again that I think every patient potentially is a research subject, and so don't assume someone will not be a subject or not interested, or maybe they're not a good candidate. Always keep an open mind on this. We need to make sure that we can meet expectations when we think about recruitment. Often we go to sites and they say, oh, I'm going to recruit 10 patients a month. And that's not really realistic. So try to find the metrics of how many patients come in that fulfill criteria for a certain study, and what the resources for your clinic to do it, and then be realistic about what you can actually meet in the study population you're trying to study here. And always, I should say it's always overestimated. So obviously underestimate what you can do. It's always better to be a bit safe on those things as well. There are a lot of instructions for clinical trials. And again, you can go to different journals and websites to see what the author's instructions are. There's the consort agreement for RCTs and randomized trials, which you must follow, and you must look at that. Design your study specifically using those guidelines, and register your prospective trial on clinicaltrials.gov. Most publications and most peer articles require this now as part of your work. It's always nice to read editorials and write editorials in journals, and it's a good opportunity to be able to, again, get published if you have a good idea or a commentary on a particular topic. Understand how ethics works. As I said earlier, what I mentioned earlier about always getting approval, and there are different ways to do this, whether it's a private or whether it's a university-based or hospital-based RAB. Sometimes it does take a while. And please know the Declaration of Helsinki that all studies we need to adhere to. So bias is an important thing. And we see bias in all kinds of studies, even what we think are high quality RCTs in ophthalmology and all of medicine. So we must be aware of the biases that exist, and we must make sure that we try to limit them and certainly identify them. These are some of the more common biases we have. A research bias. Well, I'm a researcher in glaucoma, or my particular favorite device I invented. Well, I'm biased in that. Or maybe I'm a traditional doctor. I'm a very conservative doctor, and I don't like new technology. Well, I have a bias because I'm very conservative. That's your bias. So everybody has a bias, whether it's financial or not. And often we highlight financial, but we must mention there are many, many biases out there. And it's incredible how people often have selective evidence that people use often to support their position. Selective evidence-based medicine rather than being open about it. And so just as much as financial bias can cloud the judgment, there's no question that your research interest can cloud you, or maybe someone else who you don't like, or maybe some other idea you don't like, you're going to be biased against that. And it's a big, big potential issue there. Specific study designs like selection bias, confounders are always a problem. Attrition, patient losing follow-up, and funding, again, are all important biases to remember that as well. So methodology, of course, can be think about in 4 different areas. We talk about design, sampling, data collection, data analysis. If it's inappropriate and is designed poorly, then the results of the study can be misleading. So make sure that we spend the time to define and to design the study properly, and to ask the right questions, understand, the sampling biases that can occur, make sure we collect proper data and do the right analysis. All very important as well. I mean, when we get into the methods section of a paper or of a protocol, think about all these things to think about. Define your population. What are the inclusion-exclusion criteria? What's the duration of the study and the follow-up points? And what are the details of the interventions and the examinations you do on those patients? What population are you setting off? Like I said, what are you going to include? Who are you going to include? Who are you going to exclude? Are they going to be very homogeneous? Are they going to be very different? And that's going to cause a confounder. Try to include patients as similar as you can to reduce the confounding issue. Sample size is critical because we need to have statistical power to be able to detect a difference or not. And knowing how to do a sample size calculation is important to do this. So do that properly. And again, this is also a consideration when we do studies to, of course, do things ethically and protect patients. Many years ago, I had some time. It actually wasn't during COVID, but it was during a week I had off as a resident, and I was supposed to go on vacation, and it didn't happen. Or maybe this was as a fellow. I don't remember, early in my career. And I just suddenly decided, you know what, I'm going to spend my week to basically learn how to do statistics. So basically, I went out and I got all these books, and I just started reading them. And I just read and I read and I read. And I would tell you right now, those of you who want to do something right now and you want to do research, pick up one of these books and start reading. You have time now to do this. These are some really simple books for medical clinicians that I think can help to develop an understanding of how to do research and help you with some of the different lingo that's important out there as well. Collection, again, we typically use Microsoft Excel, very simple to do that, and we complete forms as needed as well. It is helpful to get statistical people involved early, very early. Don't call them later on when you finish your study. Please don't do that. Have them help you set up your study so it's designed properly. And it's very helpful and important to get some understanding about some statistical programs. You don't have to necessarily be a coder, although these days it's easier to code, I should say. But these are packages that are available online, or you can download them. And they are really important to do anything more than a basic analysis. You can use Excel as well, but these are, I think, helpful and modalities that I think can help to have a more robust analysis. When it comes to reporting, there's certain things we want to report in our publication. The main outcome measures are important. Make sure we don't just report the averages, report levels of significance and deviations and variations, and have the right use of tables and graphs. Whenever we publish results, by the way, make sure we always show the results of all the subjects. Even if they've lost a follow-up, if they dropped out of a study, it's important that we know what happened to all the subjects, because that can be a source of bias. It's really important as well. So be sure to do that, and again, describe the subjects and the differences between them and the results. Again, there are different ways to report data in terms of how we report them, depending on whether, for example, it's a comparative study, whether the data is normal, it follows a normal distribution, whether you use parametric testing or non-parametric testing, whatever you're doing, levels of agreement or correlation studies, all different ways to study different things. Important to do this. And I'm not a statistical expert, and I certainly rely on people who are for more in-depth work for this as well. The discussion sometimes is difficult for people to write. Do not just simply put your results in the discussion. It's a common mistake. Summarize them to the question you asked. Put them in context, talk about generalizability, compare them to other published results, talk about limitations, and talk about maybe what the future may be. And don't make big claims. And be careful when you draw conclusions. Only make the conclusions you can from the results. Don't overstep the results and make them clinically relevant. Please, please, please don't use the word safe and effective. It's such a common thing, certainly in ophthalmology. We did this study in 10 patients to determine the safety and efficacy and effectiveness of this new surgical procedure. There's no way you can determine a safety and the effectiveness of a procedure or of a drug in that population, in a small population, or in that kind of design study. We're talking about big studies that are duplicated potentially, that are large enough in a large enough population over a long enough period to really use the word safe and effective. And I think journal editors are on top of this. And this is a great editorial written by a number of ophthalmology editors and Wiley Chambers from the FDA. I think everybody should read this in terms of being careful with that term. It's a very powerful term, and the FDA, of course, uses it. And we have to be very mindful of that as well. Of course, one of the culminations of our successes in doing research is publishing at meetings. It can be papers and posters or e-posters nowadays. And it always helps to start locally and work on this. I won't go into the whole process of presenting, but there's a whole art to it, of course, as well. And then, of course, we want to be published. We want to publish it, ideally. We want to be careful, of course, simply putting results in non-peer-reviewed journals, because that's not the right place to put your results in. We should be going to peer review first. You can jeopardize the publication if you publish a result somewhere else, like, for example, in a non-peer-reviewed journal. So be mindful of that. It's very gratifying to see research being published in major journals with big impact factors. Those are all wonderful. And you need to decide, of course, where to publish and where it would be appropriate. Maybe one of your societies, maybe it's a specialty journal. There are a lot of open access journals now. There are tons of open access journals, and there are a lot of predatory journals. So be very careful and seek advice from somebody who knows these journals before you submit it to someone who's inviting you to write a manuscript. It can be an expensive venture, and there can be a lot of issues with that as well. Open access is nice to use. It gets it out there faster. There might be a cost to it. The impact factor may not be great, but it can certainly publish results that maybe not make it to the high-level journals. But there's so many journals out there to send it out to that you have a lot of choices. Of course, these are some of the top journals out there with the highest impact factor and citations. We go for these, of course, if we can. But these publications often really are looking for large trials, RCTs, very clinical relevant studies, because the competition is very, very strong. Some of these journals, the rejection rate is 80% or more of papers, and it's really difficult to get into those, understandably, and it's difficult as an editor, to be honest, to reject papers. But the reality is that we have to. The competition is fierce, and therefore the level of evidence that is required to publish in them is pretty high. There's a whole process for this. It's well described in terms of the editorial board and reviewers. The different ways to adjudicate papers that when we go through this as well. And you will be rejected. You will be rejected many times. I get rejected all the time. Remember what I said before, though. Failure is not an excuse. We got to get back up and go forward, learn from the reviewers' comments, and resubmit it somewhere else for publication. Nowadays, a lot of journals have a quick turnaround time to help you with that, so you can move on. And believe me, many innovative ideas and great research have been rejected because someone wasn't interested in it or there were other issues with it. So don't be discouraged by that. Read. Publish simple reports. Do auditing if you need to as well. But whatever you do, don't get stuck. And remember, it's about the journey, not the destination. It's about the people, not the outcome. And remember that when you're going through this. And if you cherish the journey and the people you work with, I think you'll find it to be much more gratifying. So stay in touch. Any questions, please throw them my way. I am not an expert in research or a scientist, but I like it and I enjoy it and I do it. And I hope those words will help perhaps to give some thoughts to all of you. So thank you again, and happy to take any questions there may be.
Ike, thank you so much. It was terrific.
Well, thanks, Juan. You're a leader in this area, man. You've brought so much to our field. Thank you.
Listen, I'd like to start a little interaction with you, if it's okay.
Please.
And I'll start with the words from a friend of mine who's a professor of ophthalmology up at Yale, now retired. And his phrase that I love was that the joy of research comes when you discover something that only you know. And at that point of the discovery, you're the only one person in the world that has found or discovered that one thing that makes you unique. And that you're willing then to share that with the rest of the world, especially your colleagues in medicine. And having said that, one of the things about research that I find very important is ethics or ethical research. And this is where I would like to come with the exchange with you. There are papers out there, there are projects out there that are commercially oriented, and you've touched on that, meaning that a company comes with an idea. Of course, they have a vested interest or an investment in the idea, and they just want to make it work, or they want you to show that it actually works. What do you do with that?
Well, I think that we can't do research without having the right ethics, and it is difficult because you're caught in the middle often, because you want to do research, you're interested in it, and the company may kind of be bending things and turning things. And once you sell your soul, though, once you sell your soul, you're done. You're done forever, in my opinion, and you can't go back. And so be mindful of that. There are polite ways to step out of it in doing it. I think if an industry partner is smart, they'll take your ideas and your thoughts and modify and change things. But I'll tell you, I'm sure you have as well. I've been in many arguments with my co-authors or with industry partners on the results. I sometimes have to step out of the study if I'm not comfortable with it. And I can understand those that are junior and starting off, it's a hard position to be at. But your name means a lot. I mean, you built your career, you built your name on your career, and to get into something that is unethical is absolutely dangerous to patients for the first point, for patient safety, and unethical in terms of twisting or turning or maybe doing something wrong. Often industry will bend it, will push things a little bit, and they're looking for us to be pushed back. Honestly, I think the partners I've worked with, I've been lucky enough to work with most of my partners in industry, and they've been good about this and understand this. And the business side of it, I think, only works if the science part works, is what I tell them. So, but thank you for mentioning about ethics. Ethics is critical.
So, just as a matter of introduction, Maria, who's my research everything here in San Domingo, is right next to me, and she can't take her mask off because she's under surveillance right now and protection. So the next thing I wanted to touch on is bias. And I remember a phrase from Andy Corley, who was then CEO for Ionics, and we were doing the research on the crystal lens, the accommodative IOL. And he said, Juan, you know, when people invent something, it's like having a baby. You know, when you have a child, it is the prettiest child in the world, and you don't want anybody to touch it. And I'd like to know your feedback on that, because we get into pilot studies, and we've done 8 pilot studies on glaucoma, the gadgets and femtoseconds and this and the other. How do you deal with that? I mean, you have somebody who really had a great idea, invented it, but you know, there are modifications that come along the way, especially if you're involved in research and development. And it's true. People don't allow or don't want their own product to be changed, but yet there are things that make it better.
Well, there's plenty of examples about this inventor problem that we see, and it's killed projects. It's killed projects literally. And so again, I think the first thing is you just right now is recognize that bias and recognize that that challenge that we have. I think some trusted people that you trust to be able to give you advice or give us advice about a particular study or project is very helpful and very important to do that. And I think bringing other people and not just yourself is really important along for that journey. Collaborating, you know, for example, you and I may collaborate on something, and bringing you to the table, I think will hopefully, and a trusted one like you would give the proper advice to guide that process. But it can be destructive. It can be destructive because that bias is very strong. It's like I said, it's your own baby. Nothing could be wrong. And in the end of the day, it can really sink a project that could potentially have been very successful, but because of that inventor bias, it can be a problem, especially for those that start a company. It just can be absolutely terrible. So one has to know when to let go. One has to know that bias exists and challenge that. And it starts again from the very beginning. And I think the successful folks out there who have been able to do it have been able to kind of balance that that difference of that inventor bias, as you mentioned. But yeah, I certainly seen the ugly side on that part of it.
So let me touch on another one, and this is because I think what we're saying is important for the young ophthalmologists that are listening, particularly my residents. Oftentimes, when you're doing a paper or you're doing an investigation, you get into what I call cherry picking, meaning you really want something to work. You really want to show or demonstrate that some new drug or some device work, and you unconsciously start saying, oh, well, I'm not going to use that data because the patient wasn't followed. But you end up choosing that which accommodates the result that you want to happen. We call that cherry picking or research bias. But when it comes with things that are commercial, then you have companies that, for commercial reasons, also would like to do cherry picking. Tell me how you handle that.
It's a great point. We all want to show results, and we typically want to show results where there's a difference, right, or where there's an outcome. And when you see no difference, it can be discouraging. And so there's definitely ways you can massage the data. You may exclude people because you think, oh, you know what, that wasn't really a proper patient. They didn't take the medications properly after surgery, or they didn't have the right follow-up. You exclude them for different reasons, or maybe we'll change the outcome measure a little bit, right? We'll maybe change the IOP criteria for success to show a difference. There's so many ways to change things, right? To make it look better. I mean, we can dress up. You can dress up. Talking about babies, you can make any baby look beautiful, right? Depending how you dress the baby up, and that is the challenge we do face. So I think again, astute reviewers will see this, and I think they will hopefully call it out. Having a registered trial is important because that's documented now in terms of what the actual outcome measures were. But it does happen, right? And I think what you're mentioning again is what happens with industry, because people want to show things in a certain light. And again, I think it's okay to show different outcomes. That's okay, but you have to show the main outcome. You have to show all the data. And if you decide to sub-analyze something, whether you do an intent to treat or per protocol, analysis, or you choose to basically cherry pick, say you're doing that. We're basically saying we took the subset of patients, and we are now evaluating this to show this, and then discuss it in your thing. But show all the data. I can't emphasize enough. Show all the data. Let the reader decide about what that data means, and adhere to what your pre-assigned main outcome measures were, because I think it's important, because that observes the sanctity of why you did the study. But you're identifying many different potential traps with research in general, and also working with industry or sponsors. I think it's really, really great point.
So let me get on with the starting a study or having an idea that you would like to prove. And you already mentioned the most important thing is to read, or in other words, do your research and try to find out as much as possible about what you want to investigate, and then to formulate the right question. So many times, the problem that I most likely encounter in doing research in ophthalmology is the size of your study. In other words, if you're going to do something that's going to improve endophthalmitis, and you're only going to do 30 cases when the incidence is 1 out of 3,000, it's just not likely to work. So how do you go about setting out your project? How do you go about formulating the right question and formulating the right size of your sample and putting out your abstract or your proposal?
Yeah, I mean, again, you're so right. I feel sad sometimes when I see someone come to me, one of our research fellows, and they have an idea and they say, we're going to study this and compare this. And I realize in my mind as they're doing this, that to actually do a proper sample size and have the right sampling to power the study enough, they're going to need like 800 patients. And unless there's a major difference, we really won't be able to show it. And explain it to somebody is difficult unless they have an understanding of some basic statistics and sample size calculations. So I always go back to saying we need to have a sample size calculation, number one. If we do a sample size calculation, we can at least understand what it takes to power the study at the right level to have enough subjects in there. And I think that's challenging. And I think I see studies getting started all the time. For example, here's a classic one for glaucoma. We're going to study 2 different procedures, and we're going to basically look at IOP differences, right? Well, for example, depending on the standard deviation, to see, for example, 1 millimeter IOP difference or 2 millimeter difference, we're going to need hundreds of patients to do that. And on top of that, when we talk about medications being used now and how medications can, of course, be a confounder with the IOP result, then we really can't use IOP to basically judge the success of a procedure or of a certain technique. We have to look at, for example, a success criteria, a certain pressure on no medications, for example. And then that requires a different kind of analysis. And so it gets complicated. And so, I think that has to be done, though. An underpowered study is a common problem because of sample size calculation. So I think, again, it means that it's basically a level of awareness and knowledge that, understanding what power means, understanding what statistical significance means, and we can argue on what a p-value is and what's important or not, but regardless, it's our metric we use, and then understanding how to do and what goes into doing a sample size calculation. I think those are important to do that. It's nice to take on certain things, and it's okay if you can't, but then do a pilot study. Call it a pilot study. That's okay. And we're going to do a series of patients, and we're going to do, here's our results, and here's the limitation of the study. And I think, and that's perfectly fine. But don't take on more than you can. It sounds depressing because a lot of people then have a hard time to do studies. But without having the right rigor, it's really, really difficult to draw any conclusions. And then you wasted all this time and money.
I have another one that is very apropos for glaucoma in the Caribbean. And that has to do with the ethnicity in our highly pigmented patients. And I don't know if you run into that, but I'm sure you do, especially when you go to the ESCRS meetings and the European meetings, because of course they're all white or Scandinavian or Anglo-Saxon, and you try to tell them that our patients are Black or they have a different scarring reaction than those that are of Scandinavian origin or Anglo-Saxons. So sometimes trying to convince people about the difference is very difficult. And you almost have to be aware of your limitations when it comes to your statistical approach. But then we end up having to live with that population. In other words, I don't care about the data up in Scandinavia because it's not going to be appropriate using 0.2% mitomycin C when I have a patient that I know that unless they go for the 3 minutes of the 0. It's not going to work. So how do you handle that?
Well, I think we're seeing this in present day as well with some of the things that we're working on. And I think that is, again, a very valid point about patient population. It's something, again, that they speak about generalizability. And we always need to look at, for example, yesterday we had a review with our fellows about, for example, the use of Ologen in glaucoma surgery. And some studies see a difference, some studies don't. And we won't get into the details on that. But one of the differences are, for example, the study population, Asian versus Caucasian, right? And the generalizability, have to be very careful. We know that genetics and ethnicity plays a role, particularly, for example, glaucoma surgery, and for probably other things we don't know even. And so that's really important to sort of define that, understand that, and don't be careful generalizing it from one population to the next. That's why all of us have different opinions. Not only population one, of course, but also surgeon, right? For example, if it's a surgery, surgeon technique will make a difference in the outcome as well. Maybe one person's results are better than the others because their technique is better in how they do it or different. What do they use around the time of surgery? What drugs are they using around the time of surgery, for example? So it may not be even the device that's the difference. It may be, again, like you said, the ability to deal with someone's wound healing because their ethnicity is higher risk, or how they use the actual agent, for example, around the time of surgery. So I think that's why it's really important to define the population we're studying. If you can design a study with a large racial population, that's ideal. But again, it's important to have enough in each of those groups. Because one thing to say, I'm going to include everybody in the study, but if you have 90% whites in a study and 10% Black, and small Asian or Hispanic population, then we can't generalize it to the other groups, and we have to recognize that. Same thing applies for male and female, right? Same thing with male and female. This is also a common problem, for example, in other specialties as well. And there's system issues too. Some systems are more health efficient and have a better healthcare system than others, and that can also make a result in comorbidities. So you're right; these are all, I guess, what we would call confounders. And I think recognizing confounders and smelling out confounders, really important to basically identify the biases in there. And it's okay to have that, though. It's okay to have that, but that's where the limitations section in your discussion should be listing those potential confounders. It's okay to have that. Don't be afraid of saying, this is my limitations of the study. It's actually a good thing to do that, but help the reader to know that.
So one of my pet peeves in the DR, especially in my clinic and with my hospital, is that some people know the importance of data gathering, meaning that CRF, that form that tells you at week 1, at week 3, at week 6, we have to have XXX procedure studies done. And you get faced with clinicians that say, well, wait a minute, I'm a clinician. I just want to move my clinic and get rid of the patients as soon as possible. And the attention to the detail of the investigation gets lost. And I would like for you to address what it means for the FDA when you have lost a data point in a patient that you've already invested $10,000 in studies and whatnot, and what it means in terms of a catastrophe for a research project when you don't do the appropriate follow-up.
Well, I mean, Maria probably will tell you, I mean, we hate that loss to follow-up. We hate attrition, man. That just sucks, right? And of course, retrospective studies are very, very prone to this because we discharge patients, they move, whatever, we don't follow them up. But it's particularly unnerving when it's a prospective study, well designed to have a follow-up for a year or whatever it may be, and we don't meet those endpoints. That's a huge potential bias risk here. And certainly for a registration trial like the FDA, it can be a real killer with regards to the approval. And so, I think, again, that's part of the recruitment thing. Sometimes we're so eager to recruit patients that we recruit people maybe who aren't the best people to follow up. It's also important, again, so that's a problem with patient recruitment. It's also a problem from the surgeon perspective as well. The surgeon has a different mentality in terms of following up patients or being available to see patients. You know, and the clinic is not prepared for that. That's also a problem. Part of things in studies is of course patient reimbursement for their time. That does help, I think, for retention. And I think keep on motivating patients, treat them well. But it starts with the investigator, of course. If the investigator and the surgeon is not in that mindset, then the patients are not necessarily going to want to follow up. I think a good explanation beforehand about why we do research without scaring the patient away, because anybody who consents patients for studies can talk a patient to a study and can talk a patient out of a study. It's really a matter of doing the right balancing act to basically help explain them the value of being in a study. Here are the values for you, and then here's what the expectations are. And I think that's a balancing act. And you're right, Juan, again, that this is a pet peeve, and it's something that we grapple with and fight with. I mean, I'd rather have low recruitment and have good retention than have over recruit and we lose half our follow up, because that's not good data. It's actually a tarnish on the study.
I know my connection is not very good, so I may have to repeat the question. I would like for you to tell the residents what it means phase 1, phase 2, and phase 3 of studies for the FDA, because it's not something that's intuitive outside of the United States.
Sure. Yeah. I mean, you know, that terminology is starting to get a little bit outdated, to be honest. But I mean, I think we still have the same principles about what we call phase 1, phase 2, phase 3. So you can use an example as a drug or device. A phase 1 study is really just to see the feasibility of a device to make sure that it's safe to use in the first in-human study. So it'll be a small study. We're not trying to prove efficacy. We're not trying to prove some major result. We want to make sure that it's not unsafe, that it's safe to use, and we don't have any really crazy results. Often these patients are patients who are not necessarily the actual candidate for the particular device or drug, but they're a candidate maybe where the risk profile may be appropriate to take. For example, a new drainage device, we may be doing them on blind eyes because, you know, if there's a complication, the risk of a complication, the magnitude is not going to be so severe compared to somebody who sees. And that's kind of a very early first in human phase 1 study. And there's different divisions, 1A, 1B. We won't get into that. Once you establish that, the next side is to do a small study to establish some degree of efficacy, to give you enough to go on to say, you know what, now we're going to go on to the next level, which is going to be that big trial to prove the results, to prove, for example, something safe and effective, which is typically a phase 3. A phase 3 study, for example, is an FDA study, registration trial, a big study to prove something is both safe and effective. But a phase 2 is on a smaller level. For example, it may be a dosing study. It may be a study to evaluate 4 different doses of a drug to see whether there's a difference in efficacy between them. It's a smaller study. It's harder to draw wide conclusions in terms of safety and efficacy, but it's enough to give us to say, you know what, this can work, and maybe this particular dose is better, or this particular design is better, and we can now build on to the phase 3 trial. Then the phase 3 trial is the big one, okay, where, you know, you have the auditors in there, the FDA in there, proper design, and it's a big endeavor, typically a longer term study than a phase 2 study would be, and with specific outcome measures that are defined based on the phase 2 results. So phase 2 results will give us an idea what the standard deviations will be, what the metrics are to measure, what adverse events we should be measuring, and then we build it into the phase 3 study. And then that's basically the studies you have. Phase 4, we talk about, like, for example, post-approval. Maybe we have a drug that's approved, or a device that's approved, and we haven't studied it properly, for example, in pseudospallation glaucoma or in Black patients. So we'll do a phase 4 study to say, you know, we're going to do this study with an approved product in this new population, or maybe with a new dose or new technique with a surgical study to show it works or not. And it's going to have a post-market study. Often the FDA mandates companies to do post-market approval studies because they want to be able to show that it's okay in a particular group. And it can often be done to change labeling for a study for a particular product, because the labeling will be based on the phase 3 design. This is approved for use in combination cataract surgery from mild to moderate glaucoma to lower pressure. But maybe we'll do a post-market study to maybe change the labeling a little bit to open it up to maybe different populations. So that's kind of a long answer, I guess, for our short abbreviated version for the question of phase 1, 2, 3, 4. I hope I answered it. I hope I was right, Juan. Was it okay?
Yeah, it was great. There's only one thing that I would like for Maria to address the residents about what it's like to deal with our FDA, which is the Bioethics Committee in the DR. And I'm just going to pass the camera and the microphone to her. So Maria, take it on.
¿En español o en inglés?
In English.
Okay.
Hey, Maria.
I'll be right back.
How are you, Dr. Ahmed?
So Juan is going to do a cataract. He'll be back in 2 minutes, right?
Yes, that's right. Dr. Bai is talking about our regulatory authorities, but I wanted to add my challenges in time with clinical research, and one of them, and I would say it is one of the most important, is to earn the respect of the doctors in relation to data gathering. You have to do that. Okay, later on, when the patient comes back, no, you have to do it now. Yes. That is one of the most difficult parts. And also, when you have a big company that is wants to do a trial, like let's say Abbott, Johnson and Johnson, they have a lot of resources. But then you have startup companies that have such a good product or idea. They want to do some research, but they don't know anything about research, but they do know a lot about their product. That is an extra burden to the clinical sites, because you have to be the site, but you have to add ideas, provide ideas, and it is difficult sometimes. I don't know if you understand.
I completely agree. So both points are very, very good. Sorry, did you have something else? I didn't cut you off.
Excuse me?
Did you have any other questions? I didn't mean to stop your question. Sorry.
No,
I'll answer your questions.
It was not a question. Yes.
Well, I think so. Before you go there, Maria, let me take 2 points, and then I didn't want to interrupt you. I didn't want to be rude, but I want to just quickly answer those questions. So data gathering, yes. What I think helps for residents and fellows is to understand why we're doing the study, first of all, because sometimes people don't understand, why are they asking for this data? Why is it important? Why do I have to measure this thing, for the patient who's doing so well, right? But helping to appreciate why we're doing the study, what it's about, the product may be what it is, what the potential benefit may be, at least starts the doctor to understand why we're doing it. The other part of it is understand the biases in studies, right? And I think when residents understand the bias of study, for example, not gathering data properly and what the implication is, I think that may give it more respect. So even having discussions like this, for example, where I think understanding what the biases can be by not doing proper data gathering may be appreciated. I will say that there are some doctors who really love the research and some who really, run away from it. And the ones that run away from it, maybe they run away from it for the wrong reason. And I think research does not have to be complicated. And I think any level of interest, I think, can help the motivation part of it to gather data. I think it's part of any clinical exam, for example. We wouldn't want to treat glaucoma without doing gonioscopy. Well, we don't want to see the patient without doing the proper data to gather for that particular one. It's also sometimes happens inadvertently, like the patients come in, they don't realize they're a research subject, right? Or they didn't know they have to measure certain things. So having reminders from staff, sometimes our research staff have to be right in there, right on top of them, because it's a busy clinic, everything's going on, there's a ton of people in the waiting room, emergencies happening, right? So having someone to help you out, to remind you, to guide the patients, maybe even having a separate examination room just for them, you can go there quickly and come back, set it up for you, right? Because, and knowing ahead of time, saying, Dr. Baye, today you have 50 patients in the morning, and these 10 patients are research subjects. I highlighted them for you. Be prepared so they know, rather, so it's not surprising. So that at least will help the day go better when it's there. The other thing I'll mention is that when you book research patients, book more time for them, right? They need more time. So don't book them for a 5-minute slot for routine check when they may need longer time. So I think all those things we try doing to help it, but I admit it's sometimes a challenge. And I think those are some pearls on that. As far as the company stuff, yeah, I can think of a few products recently where a company dropped off a device and they say, okay, study it, right? And we know more about it than they do, And it's like, oh my God, right? So in some ways I'm like, what the hell, right? Are you kidding me? On the other hand, I'm thinking, well, it's an opportunity for us to really, lead it, right? So in some ways, sometimes I like it, but if the company is not in tune, then of course we cannot build a device, we cannot change it necessarily. So that's where it gets frustrating. So I think that's again where we don't work with everybody who wants to work with us. We are selective. We turn down if we feel that, you know what, this device is not really worthwhile,
Yes, no lo llamo.
the knowledge is not very good. I think those are all things that that we think about on there as well. Good question.
Oh,
Perfect.
what I just wanted to thank Ike and just say one last thing. One of the neat things about doing research, and it goes with your answer about some people scaring away from it, or I personally have benefited a lot in terms of my own knowledge and my own skills simply by doing research. Why? Because I've learned things that I had never been done before, or tried things that have never been tried before. And you have somebody looking at you saying, hey, you're doing this right or you're doing this wrong. And this sort of autocritical self-editing part of research, I think, makes you grow as an ophthalmologist, as a professional. So for those of you that are shying away from research, let me tell you, there's a lot more to be gained from it, not only in knowledge and skills and just satisfaction after what you seek. So Ike, thank you so much. I think you're going to be an inspiration for many people.
Well, Juan, you're an inspiration for me and for many around the world. And I remember meeting you, one of my first meetings at Bascom Palmer, and the knowledge you bring and the attitude and the you know, the passion, it's incredible. So you know, and we all know how instrumental you've been in being a first user for many different areas. You've guided products to be produced and used in millions of people around the world. So I think Dominican and the world are very proud of you, and we're proud of you. And I'm honored to be part of this discussion. And you're a great friend, my friend. You're a great friend. I appreciate it. And Maria, thank you. William as well.
Thank you.
Thank you for all of this. And I keep my prayers and safety for you and health.
Amen.
Thank you.
Thanks a lot for your time.
Thank you, everybody. Keep well. Keep in touch.
Hey, Dr. Ahmed. All right. Ok, le enviaremos las preguntas personalmente al Dr. Ahmed, las preguntas que hicieron vía YouTube y las que tienen vía el chat, para ver si él, como él nos puede dar una respuesta un poco más adelante de lo que habló el día de hoy. Quiero agradecer a todos nuestros attendings, al Dr. Valle, a William, por haber facilitado la llegada del Dr. Ahmed a nosotros y por haber hecho un gran trabajo como anfitrión. Muchas gracias, William, y gracias a todos.
Gracias.